Right, so let's get started. I'm going to move some stuff around so that I see what everyone else sees. So my name is Niek Buurma. As mentioned in the introduction, my interest is in reactions and interactions in aqueous solutions. In order to understand, let me also switch on the laser pointer. In order to understand where that interest comes from, you probably have to know a little bit about me from a very long time ago. This was me being found by my lab manager. I normally call her mum, but found by my lab manager, who realized that I was not wearing my lab coat while doing my first experiments with water, as you can see here in the background, with a very perceptive audience. Now, a little bit later than this, I started university and I started to study chemistry. And this particular person was my organic chemistry professor. Now, you may recognize this person because this is the person who years later would win a Nobel Prize for the development of a molecular car. And here you see a structure of that molecular car. So, okay, that's great. Win a Nobel Prize is always great. But let's have a look at that particular molecular car. So this is a 3D picture of that car. And this is what that car looks like. And it is a single molecule car. Now, it's a great molecule. It's an enormous amount of work to synthesize this. And as you could see on the previous slide, it does actually drive. And it drives something like 6 nanometers. So this is that molecule, and it's a molecular car. But now let's have a look at a real car. And for those of you who have forgotten what a real car looks like, it may look something like this. This car is clearly not a single molecule. It is built from components. So the question that we are now asking ourselves is, if real cars with functionality to drive us somewhere are built from components, shouldn't we try to do that on a molecular scale as well? Because although this is an absolutely amazing molecule to synthesize, you would prefer, it would be a lot easier if you could simply take the blue wheels, the chassis, the motor, and put them together as you would in a normal car, so that the car is assembled from components rather being synthesized in one very long synthesis. So in order to do that, what we would need is molecular scale control. We need to know which component goes where, and we need to know that for multiple components, and the components need to go to the place where they need to go, a little bit like the blocks in the cartoons over here. So if we were to find a way of making sure that every component knows where it needs to go, we would end up being really rather happy chemists. So that leads to the question of how we actually build. And when we build materials, when we build structures, normally we start with a set of instructions, a set of components, and we then put everything together to give us an object with a certain function. In this case, a table to put our newspaper or our coffee on. So what we need to find from the macroscopic world, we have instructions, we get them, for example, as a little booklet, and then we know where to put the components. So how does that work on the molecular scale? Well, on the molecular scale, what we have is definitely not our hands and an instruction booklet to put different components in the right places. What we have are molecular vibrations, Brownian motion, and that's it. So normally, if this is a live lecture, this is a little bit easier, but I'm going to try to do this now by switching or by stopping my sharing for a second, so that hopefully I should become full screen. Imagine you have this little bit of, oh, there's a green brick in here, so that's why one of the bricks is invisible. I had not anticipated this effect. Let me just take that green brick out. Imagine this little Lego structure over here. It's actually quite easy to construct. So I have a base plate, red, orange, yellow, and blue. If I have the right Lego bricks, I know where these need to go. So I just put them together. What was the next one? Yellow. And oh, the green one. And there you go, I've created that little structure. Now, if I only have Brownian motion and nothing else, it's the equivalent of taking all of these components and putting them in a little box, taking that box, shaking them, and having a look inside to see whether that structure has spontaneously formed. Well, I can do this, as you can imagine, really for a very long time. And the chances of actually finding this on the time scale of this particular lecture are vanishingly small. And the reason for that is that these bricks don't know where they need to go to form the final structure. So in this demonstration, the idea is essentially that my shaking is your Brownian motion, is the molecular vibrations. That is what you're trying to use to put the components in the right place. So essentially what this means is that I buy my flat pack furniture from a well-known shop. I put everything into my house and then I shake my house until the furniture is actually all constructed. That's going to take a very long time. In fact, it's never really going to happen. So the question is, how are we going to build without touching our building materials? So this is where we are inspired by nature, and this is a self-assembly project that my partner and I engaged in slightly over 5 years ago. This is Soren, which stands for self-organizing and potentially, that's up to him, replicating entity. He self-constructed during 9 months under aqueous conditions, the kind of typical biological conditions, but in a very supporting environment. And now I don't want to compare my partner to a wonderful high-end material, but it is rather amazing to see that babies are so well protected and fed and cared for while in the womb. The key thing is, nobody touched this baby. Everything in that baby put itself in the right place. So clearly, nature is very, very, very good at self-assembly. Now, the reason that nature is good at self-assembly is because it has an amazing manual, and those are the codons on DNA. So what you see here is an RNA sequence, which is of course transcribed from a DNA sequence, and the bases on our DNA code for the different amino acids that are going to form a peptide, a protein, or an enzyme. For those who like the detail, this is essentially the genetic code as well. You can read that, for example, GCC codes for an alanine. So we have a really good instruction manual. The way that DNA is then read is you have transfer RNA that has an anticodon. It recognizes the 3 base codons on the DNA, and it then takes an amino acid indicated by this little ball over here, and it puts it on the messenger RNA, and it puts the amino acids in a sequence so that you form your proteins. DNA is also a very robust manual, contrary to RNA. As you can see over here, under kind of standard neutral conditions, it would take something like 100,000 years before you hydrolyze the phosphodiester bond that forms the backbone of the DNA. So DNA is really very, very robust. Oh. It also has a very high information density, and all of today's global storage needs, apparently that is 10 to the power 22 bits, would fit in a box of 10 by 10 by 10 centimeters. And in 2040, you would be able to put all of the world's knowledge in 1 kilo of DNA. So if we assume that the density of DNA is more or less the same as sugar, imagine holding a 1 kilo pack of sugar containing all of the world's information. That's how high the information density in DNA is. Going back to my son Soren, he was coded by 5,000 or almost 6,000 million base pairs which is a bit of DNA about 2 meters long. I don't say 2 meters long, but that doesn't make any sense because that would make my head 2 meters wide as well. And 10 to the power minus 12 grams. So a tiny bit of DNA, his first bit of double-stranded DNA was just 10 to the power minus 12 grams. And that coded for everything that he was when he was born. Now, it's not only nature that can make synthesized DNA, we can do this chemically as well. And you see an example over here. It's typically done via solid phase synthesis. And it's essentially a protocol that allows you to couple the bases one after the other. So you can synthesize DNA with a very specific sequence. And that's going to be important for some of the stuff that is going to come up next. So the key thing that we've seen so far is that we have a set of instructions, and those instructions are DNA, molecular instructions on the basis of DNA. And that is going to give us an opportunity to engage in some biocompatible building. Now, before I go to the biocompatible building and before I'm going to talk a lot about DNA, there are lots of different types of biomolecules, and they all play a role in biology, of course. There are lipids that allow us to make compartments or cells. There are carbohydrates that can give structure or recognition motifs. There are the nucleic acids that we're going to talk about a lot in the next half an hour or so. There are proteins, for example, this particularly nasty protein, which is the spike protein in the SARS-CoV-2 virus that's going around at the moment. But another key important molecule is a molecule of water, because most of the interactions that we're going to talk about, for example, the formation of duplex DNA, is driven by the fact that these molecules interact in aqueous solutions. Okay, so how can you use DNA in directed assembly? So the way that DNA is used in directed assembly is through base pairing recognition. And most people will know that G pairs with C and A pairs with T. And that means that if you have lots and lots of bases, and then you have the complementary strand, you can essentially bring two strands together like a very long zipper. A little bit like this one over here. It is in fact the one on the screen over here now. And you can see that you can have a pretty long zipper that you can open, but more importantly, you can put it back together and you form your duplex DNA. Right, let's get rid of that. Gone. So this is how we bring two strands together. We bring two strands together selectively. And the selectivity comes from the next slide. If I have two bits of strands of nucleic acids, if they are perfectly matched, I form my duplex. But as soon as there are mismatches, for example, here an A that is going to be opposite a G, or a T that's going to be opposite of a C, the duplex is not formed. And that means that we have all that information that we have in the DNA, we can use that to bring strands together to start to form structures, more than just the duplex. And the kind of structures that you can then start to form are, for example, this cube over here. So this is work that started off with Ned Seaman, who designed sequences so that you have a red sequence that goes around the face of the cube over here, and then a blue sequence that defines this phase over here. And by cleverly choosing complementary strands, he can create a set of strands that form this cube if you just throw all of them into an aqueous solution. So it started off with cubes, and then it went to tetrahedra. So here is a tetrahedron. And again, you can see that there are bits of the strand that recognize each other and eventually fold so that you get a structure like this, or an interaction pattern like this. If you then ligate it, which essentially means you form some bonds between the strands of DNA, then the structure automatically becomes this three-dimensional structure. So we can use the information on DNA to design bits of DNA that give rise to three-dimensional structures. And that can go further because you can then also start to fold a very long single strand using staples. That's a technique that you see over here, where you start to design mats so that you bring these strands together. So that is what has happened here. you can start to make mats, you can start to weave together tiles. And what that allows you to do is to make structures like the ones that you see over here. You can make a simple square, you can make a rectangle with a loop, a star, a smiley face. And if you then look at these structures using electron microscopy, you can actually see that the information that you have put into the DNA that was designed to give rise to these particular structures is actually being used, and it gives these particular structures. Lots of smiley faces because the experiment, of course, worked. But these, so far, were all flat structures. You can go a step further by making sure that you ask your clips to create curvature to compress the DNA. You can then get curvature in these structures, so you can start to make circles in your mats, as you see over here. So you can go round in a circle, or you can make square edges with rounded square faces with rounded edges. You can really start to design more and more complex structures. Again, still in 2D. You can expand this to three-dimensional structures as well, and that's what you see over here, where you can make half spheres, full spheres, rugby balls, or even small flask-like structures like the one that you see over here. And again, electron microscopy confirms that you can make all of these structures. You can then also, as soon as you can make a series of planes, you can put those together and you can make a box. Now, making a box, is that interesting? Well, just making a box, of course, is not interesting. But boxes become interesting when you can put something inside the box, and if you can then open them to take something out of that box again. Now, the other thing that is absolutely amazing about this particular box is that you can coat it on a little bit of plasmid DNA. And what that means is that you can incorporate it in bacteria, and you can then have bacteria create these boxes for you. Because, of course, bacteria contain all the molecular mechanisms that you need in order to reproduce this DNA. So you now have bacteria synthesizing DNA boxes for you. Does the box exist? Yes, the box exists. As you can see here, cryo-electron microscopy and small angle X-ray scattering are all consistent with the formation of this particular box. And the box is hollow on the inside as well. Okay, so that's one thing. We have structures that are made completely out of DNA. And we've seen that we can make flat structures and we can make three-dimensional structures. But what we can also do is we can use the DNA and connect, for example, fluorophores, chromophores to the little bits of DNA. And that is what has been done, for example, in this particular paper. There are oligonucleotides that contain a fluorophore. We have a little bit of DNA, strand A, and it forms a duplex with strand B, but strand B contains this particular fluorescent molecule. But then the little bit of B that is a bit too long can form a duplex with strand D, which also contains a fluorophore. But that can then also, that overlaps with strand C. You continue E, F, G, H, and I. So what you can do in this case is you can put all these different chromophores together. And then there is something called resonance energy transfer, fluorescence resonance energy transfer, or FRET, that allows you to put energy into this particular chromophore, and the energy then just hops from one fluorophore to the next, and it is conducted across the molecular wire that you have now created, or the photonic wire that you have created. That, to be honest, was why I used this particular series of Lego bricks before, because basically if I were to excite a blue chromophore, energy could then jump to the green, to the yellow, to the orange, to the red chromophore, and be transported in much the same way as energy is transported along a series of different chromophores on this particular self-assembled, of course, template. You can also use this to direct nanoparticles. That's what you see over here. You can make these mats that I was talking about that have little bits of DNA that are hanging out, that are single-stranded bits of DNA. You put a nanoparticle in the middle, and then these little bits of mats will recognize each other will click together and form larger structures. And then by adding the right connectivities, again, using the sequence selectivity of DNA, you can make different little connecting balls. And what you can then do, of course, is try to recreate this famous image, but using nanoparticles. And again, well, if you are happy enough that this is an accurate molecular reproduction of this image over here, then of course, this is a very successful experiment. But in general, just being able to create lots of these, as you've seen over here, again, self-assembled, no human touched this, everything knew where it had to go, is really quite impressive. You can also use this in molecular sensors. So here is an example where we use an aptamer, so a bit of DNA over here. If it binds to an analyte, something that we want to detect, we form this particular structure. This structure then offers a good hydrogen bonding interface with this species over here. Now, why is that interesting? It's interesting because if you build that into another DNA-based tile, as you see over here, if you add, in this case, it's GTP, which is being analyzed, it binds, it brings those two faces together, and it gives itself a kiss. And that's what you see here. Most of these are closed, indicating that we have indeed given the kiss, or we're in the process of giving the kiss, because GTP is present. This one hasn't actually picked up a molecule of GTP, so in this case, it is open. Okay, but so far, everything that we've seen are structures. They are boxes, they are bottles, they are mats, and they don't have a lot of function yet. So the next thing that we're going to look at is actually a race towards making a machine, to making something that actually self-assembles and then does something else. So we'll go back to that box, and one of the nice developments with that box is that you can put, again, 2 chromophores on there, and we tend to use chromophores because it allows us to use light to study what is happening on a molecular scale. We put 2 chromophores, one here and one here, and if that box closes its lid, the 2 chromophores are close to each other. And the way that you can then see that is, again, whether you get any threat, whether you get any energy transfer between the 2 chromophores. You see that over here, if you have unlinked faces, so if you don't have the bits of DNA here and here that close the lid of the box, then it doesn't really matter whether you add the key or not. But if you have the constructed box, you can now add a key, and the key is essentially a bit of DNA that is complementary with this strand over here. So it displaces the shorter strand, as you can see in this picture over here. It then opens up the lid. The 2 fluorophores are further away from each other. So you now start to see that the fluorescence, if you excite, let's say, the green fluorophore, you no longer see the fluorescence from the red fluorophore that was brought about in the closed form by energy transfer. So you can see from the UV visible spectrum, or from the fluorescent spectrum, I should say, you can see that adding DNA opens the box. And then if there is something in that box, you can allow it to leave the box and do something else. So you could consider maybe this as a little pill box that releases the pills that you want to release, the pharmaceutically interesting molecules, upon the addition of a key that opens the box and releases the molecules. You can go a step, pun not intended, further by taking a bit of DNA that walks across a surface. So this is work by the group of Turberfield and co-workers. And what they do is they have a bit of DNA that is connected with a complementary strand. The green bit of DNA is fixed on the surface, but if it is in the form of a duplex, an enzyme can cut the green strand. But if the green strand is cut, as you can see over here, the pink strand wants to move over to the next strand that is still intact. But then it forms a duplex again, and the enzyme that the authors were using will only cut the DNA if it is a duplex. So then this bit of DNA will be cut, and this process will continue. So as long as you have a series of green bits of DNA standing next to each other, and the pink strand should lead, well, formation of the double helix with the pink strand should lead to a cut over here, and the pink strand should then be transferred to the next complete strand. And that happens. It's a bit difficult to see over here, but you can see that with time, the bit of pink DNA, it starts on stator number 1, so right at the start of the experiment, and it then walks over. Oh, I remember that my camera is mirrored. It then walks over to the other side until at the end of the experiment, it is predominantly over here. Again, in this case, surface STM, I think it was, allows you to see that as well. We can then use you can turn this concept into a DNA synthesis machine as well. So what you see over here is we have a coding strand, and then we add one after the other reactants that are linked to ever increasing length of DNA. So strand 1 brings the black reactant next to the white reactant. They carry out a chemical reaction. We form this molecule over here. We now add a next strand that is carrying the red reactant. This is a longer strand. It will form a better, a more stable double helix with the coding strand. So it will kick this strand off. This one will take its place. Red dot next to the white and the black dot. Chemical reaction happens. Add even longer DNA. That will kick off the shorter bit of DNA over here. Places the blue reactant next to the growing product over here. Get another chemical reaction, and that continues. So what happens here is the DNA is bringing reactants together consecutively so that we carry out a chemical reaction in a completely coded way where the code is on the DNA. Now, another part where you can use DNA, so all of what we've done so far is using DNA and having stuff completely connected to the DNA covalently. But what we can also do is have things bind to DNA. And if you look at the structure of DNA, you will see a phosphate backbone, you will see a major groove and a minor groove, and you have space between the base pairs. And that leads to recognition motifs in the minor groove and in the major groove. And the way that nature uses this is it recognizes the base, the hydrogen bond donors and the hydrogen bond acceptors at the bottom of the major groove using so-called helical turn motifs that put alpha helices in the major groove of DNA. So yeah, typical names are the helix turn helix and the helix loop helix over here. What we do is we use a slightly less evolved way, I would say, of binding molecules to DNA. And you see that over here. We typically use, or humans typically use, intercalators or minor groove binders. And recognition of DNA by just sliding stuff in between the base pairs is less selective. And the same is true for binding in a minor groove. There is less selectivity involved in minor groove binding than there can be in major groove binding. So that was why I was saying nature does this right. It uses hydrogen bond donors and acceptors on an alpha helix to recognize hydrogen bond donors and acceptors in the major groove. Humans are really poor at sufficiently developing hydrogen bond donor acceptor arrays so that you get perfect recognition patterns. So a key example of molecules that can recognize DNA reasonably well are Dervan hairpin polyamides, and they bind in a minor groove in a side-by-side manner. So you have two of these molecules sitting next to each other, or if you connect them, if you connect the two molecules, you get a so-called hairpin. So that's why they're called hairpin polyamides. And they can recognize up to approximately 5 base pairs. They're polyamides, so you don't see a lot when they bind. But you can, of course, change that, and that is what was done by the group of Burley and Glasgow. And they used the recognition ability of these hairpin polyamides, but they then put chromophores, or they connected chromophores to the hairpin polyamides. And if you do that, you can take a sequence of DNA and you add, so this is no longer covalently attached, this is non-covalently attached. You add the polyamide that recognizes the green bit of DNA and puts the chromophore exactly where it needs to be. So you now use your DNA structure as a scaffold to put chromophores in exactly the place where you want it. And again, this was used to transfer energy along a sequence of different chromophores, a so-called spectroscopic gradient. Similarly, you can use a self-assembled catalyst. And this is work from the University in Groningen, where people use intercalators to put a copper center in proximity to the DNA. And then if you have a prochiral reactant, the fact that the copper is now in a chiral environment provided by the DNA means that you can actually get enantiomer selectivity from your reaction. Okay, so almost towards the end, what is it then that we do in Cardiff? So in Cardiff, we take the same step and we looked at how do we organize molecules. And now, having spent a lot of time with children, of course, these kinds of toys became important again in my life. And what they made me realize is that when you ask a child to organize a set of blocks, what you use is the fact that the shapes of these blocks are really very, very, very different. And that's essentially what you want for your organization of molecules as well. If you really want to make it selective, you need to go for as diverse structures as you possibly can. So we need a molecular equivalent for this, and the molecular equivalent that we're going to talk about is still based on DNA. Now, we've worked with lots and lots of duplex DNA binders with some random metal centers in there. These ones bind to DNA. Another cobalt complex that binds to duplex DNA, which we use for in the development of biosensors. But we also know that there are molecules that selectively bind to hybrid DNA-RNA duplexes. So this is not a DNA-DNA duplex, this is a DNA-RNA duplex. And it says many other nucleic acid structures are available, and that's true because you have lots of different quadruplex structures as well. And we also have molecules that bind selectively to quadruplex structures. Now, if you look at these different structures that we've looked at, you see that they are beginning to be very, very different. Duplex DNA and quadruplex DNA have very different structures. A bit like what I said, I needed in order to sort my blocks earlier on. So there are the quadruplexes, there are duplexes, there are also triplexes, there are I motifs, there are lots of different duplexes beyond the normal duplex DNA. So this is useful. The other thing that is useful is that the information density of these structures is still really rather high. So if I say that 3 bases, 1 codon is the information carrying bit of DNA, if I look at a bit of duplex DNA, that would be maybe 12 base pairs. A quadruplex could be something like 12, 16 base pairs as well. So in terms of information density, I don't lose more than an order of magnitude in information by looking at nucleic acid structures, such as quadruplexes and duplexes, rather than looking at the nucleic acid sequence. And the other advantage, of course, is that the biological machinery for copying DNA exists. So I have a way of scaling up the production of the structures that I want to make. So this is what we want to do, is we want to identify molecules where one molecule binds to duplex DNA, another one binds to quadruplex DNA. And as always, we want to test whether we are successful by looking at energy transfer from one molecule to the other. And what that means is that we need to find molecules that have selectivity. One molecule binds to the quadruplex, but not to the duplex, and the other molecule binds to the duplex, but definitely not to the quadruplex. Otherwise, our assemblies become a mess. So these are the types of molecules, these are the pairs of molecules that we're trying to identify. Now, in a lot of cases, this leads to too many options. And for those of you who are particularly interested in machine learning, this is the kind of equilibrium system that we have been trying to Someone's car alarm goes off as well. So if it's not people angle grinding. Okay, now all windows are closed. This is going to go wrong. Pressure is building. So a ligand can now, or a DNA binder, can bind in lots and lots of different orientations, which means that we have a lot of equilibria to analyze. And this is one of the places where we have accidentally found ourselves doing machine learning to try to analyze lots of complex coupled equilibria to figure out where molecules actually go if they have lots and lots of different options. That's the data analysis part of this. What we do in the lab, or one of the experiments we do in the lab, is what we call a double competition dialysis experiment. And briefly, what we do is we take a few molecules, typically colored molecules, and we put them in the bottom of a device. And then on top of that, we have a little bit of dialysis membrane. And then on top of that, we have 5 holes. You can see them over here. And we put different DNA structures in the 5 different holes. And then the DNA structures are too large to go through the dialysis membrane. So they can't go from the top of the device to the bottom, but the molecules can go from the bottom of the device to the 5 different wells. And if you then leave them for a few days, they will go to the structure that they prefer. And what that means is that the ligands, or the DNA binders, are telling us what they actually want to bind to. And this then allows us to find out how we can identify these kind of pairs of molecules where a binder goes with one structure and not with the other. And this is ongoing work in the lab at the moment. So at that point, we'll go back to functions. So where should this go? And at the moment, you've seen that a lot of this is related to wiring, or to energy transfer from one point to the next. So why might that be useful? Well, for example, if you have a tiny molecular structure that you want to swim towards the light. So you want it to take the energy from one point, the energy of the light, but to use it to actually kick its leg to swim towards the light. So you would have to transport energy from one point of the molecule to the next. Whether this is a molecule or whether this is a slightly larger structure, it could be a nano robot. At the same time, we can make functional materials, materials that respond to their environments. And you've seen an example of on-demand synthesis, where we make a molecule completely coded by a structure of a DNA strand. So as a summary, assembly requires instructions. DNA carries information, and we're in directed assembly, we're using the information carrying ability of DNA to develop structures from functional components. And that means, in this case, that the DNA instructions plus the molecular components can lead to nanoscale functions. So we go from instructions, components, and functions. And if there's anything that you need to take away from this message, it is that for directed assembly, we need instructions, we need components, and then we get function. And at the end, I'd like to just briefly highlight what I think is the research strategy most applied in my group, and it's probably best illustrated by this particular cartoon. And I'll leave you to read that, and I'd be happy to ask questions as well, but also to answer any questions if there is still a little bit of time left.